The session on the C. difficile vaccine. My name is Catia Ferreira. I'm the global medical lead for Pfizer vaccines, in particular working on C. difficile. So it's a real pleasure to be here, and thank you again to the Peggy Lillis Foundation and Christian for inviting me. If you want to see the slides slightly bigger, just go ahead and press the screen twice. Just tap it twice. And you should be able to expand those slides. Now, if Kristen, if you want to give me a thumbs up, if I can go ahead and present, that'd be great. Great, thank you, Kristen. So I wanted to take this session to really talk to you about what we consider important or how we go about thinking about when we want to develop a vaccine. And I will use C. difficile, of course, as an example, but really the principles apply in a lot of our vaccine design and how we think vaccines play an important role in disease prevention and in general, keeping us all healthier so that we can live longer, healthier our lives, and we can enjoy and age appropriately as we go through the continuum of our lives. Now, if I can go to the second slide, please. So for C. difficile, I don't think I'm going to tell you anything new here, other than when we consider a vaccine or when we consider to develop a vaccine, we must first understand how the bacterium, or in some cases the virus, or how the effector causing the disease, in this case C. difficile bacteriums, work, and what is the mode of action by which they cause disease. And in this case, C. difficile is a bacteria that expresses toxins, in particular two toxins, A and B, and those are really responsible for the majority of the C. difficile disease that we see today. Now, other important aspects to consider while developing a vaccine is what leads to that disease. What is it that it marks a group of individuals that might be developing this disease? And for C. difficile, again, the important piece in here is the high use of antibiotic really marks those individuals as the biggest risk factor for developing CDI. So when we looked at how we develop our program, in particularly, we wanted to understand how antibiotics play a role and how they play a role in disease development to make sure that we had a chance at understanding how might we go about preventing disease. Now, the burden of disease is really important. Obviously, we want to focus on developing vaccines that will help populations at large. That's the biggest goal that we have, is a public health importance. And for C. difficile, and this year we just got the reconfirmation that the burden of disease for C. difficile has really continues to be a very high and heavy burden of disease. And we see improvements in the healthcare acquired infections. We do see a reduction in cases, but overall, the community-acquired CDI cases continue to increase over time, and we continue to see a high level of CDI cases in here in the latest publication in 2017 by the CDC and its EIP surveillance. We reconfirmed that there are 462,000 cases of CDI, with over 20,000 deaths in the US in 2017 alone. And that's really important. It's a large burden of disease, so there is an extreme need to build a preventative measure that hopefully will continue to help reduce those numbers in the near future. Now, finally, it's important as we develop our vaccines and we have commitments with our governments, our Centers for Health and Disease, that we develop vaccines that are for extreme needs and urgent needs. And CDI or C. difficile infections have really been declared a public health threat that require urgent and aggressive action as per the CDC. And in Pfizer, we agree. We believe there is an important space where today there are no vaccines to prevent any initial CDI cases or recurrent infections, that there is a big gap in prevention, and we would like to try and help solve for that gap. And this gap is not just in initial prevention, but also in the space of potential future treatment failures and recurrent CDI, where perhaps a vaccine strategy may be able to help. But right now, where we are in our program, we're really focused on preventing CDI before it occurs. That's our biggest goal. So next slide, please. Now, why might we need a vaccine? And I've already alluded to this. When we go through our continuum of life, we notice in a lot of our infections, or in a lot of infections that occur to individuals such as us, that age really plays a change over the period of time. And as we age, we become more susceptible to diseases. We see these with other infections, but CDI is a good example of this curve that we see of an increase in CDI over the age period, because our immune system starts to work less effectively. Our microbiome, which is a big portion of how CDI interacts with us, changes, and for individuals as they age have an increased likelihood of contracting CDI. Now, we also understand that although traditionally the first cases that were really discussed and the first epidemics talked about CDI really lived at the hospital setting, so there was a big focus in understanding hospital-acquired infections, understanding how might we prevent hospital-acquired infections. What we see today, as we learn more about this bug, is an increase also in the community level infections. These are individuals who are not necessarily in hospital, who might be in our community, who might be leading very traditional, normal lives, but then present with an increased community likelihood of developing CDI. And I think I wouldn't be out of place to say that a lot of us who are here in this call have seen these cases of community CDI really increasing, and the CDC really points to an increased burden in community CDI to go along with the high burden of hospital-acquired infections that we see today. Now, it is likely that those most at risk, so those, and when I talk about risk, I talk about really a combination of antibiotic usage, healthcare associated interventions. All of those may point to a population that may benefit from a vaccination against CDI. So in a future world, if we were able to understand who might be susceptible to develop a disease and who might be at risk of in the future potentially coming into contact with CDI and developing the causes or the effects of a C. difficile disease, we might be able to then vaccinate them and prevent them against this terrible really terrible disease that we see today, and that's why we are here talking about it. So next slide, please. Now, finally, at Pfizer, and this is a really part and parcel of what we do today and what we represent when we say we work for Pfizer. In our vaccines department, and certainly across the company, vaccines for us represents an opportunity to continue help improve health of individuals across a life continuum. We believe that vaccination is one of the greatest public health advancements and one of the greatest interventions that you can have beyond just the largest of interventions that continues to be the access to clean water. Vaccination has prevented vaccines across the centuries and really reduced large burdens of disease. And I believe truly, and today in the current epidemic that we're suffering today, we can see the real power of vaccination and how it might help prevent disease. So the preventative health power of vaccines are long recognized in all populations of all ages, including from pediatric to adolescent disease, but also in our elderly population. As we live longer, we want to continue living active, healthy lives. We want to prevent serious illness in diseases in adults and in our elderly, so that we can continue to contribute to society and, frankly, continue to contribute to a healthy, happier, longer life. and therefore Pfizer is committed to continuing the development of new and innovative vaccines to help prevent infectious diseases globally. So CDI, a C. difficile vaccine, is really one of our many candidates that we have in our pipeline that we believe will have an important public health impact and hopefully a very personal impact to a lot of us here in the session today. Next slide, please. Now, just to reiterate, in the US, and in particular the population at large in the US, when I look at the numbers, CDI or C. difficile disproportionately affects older adults. This is not unusual. This is an expectation given that as we age, we have more contacts with health systems. We are more likely to receive antibiotics. Our immune system is changing and our microbiome is changing as well. So as we age, we see this increase in CDI risk over time, which is really what that graph is pointing to you. And where you see the real largest burden of disease is in individuals 65 and older. But saying that, although the largest burden of disease, which I believe is a number of 600 over 100,000 population here, remember we have a 300 million population, 100,000, within 100,000 individuals, 600 of those will develop CDI if they're 65 and older. When we look at the younger cohorts, there's also a significant burden of disease. So it's really, we feel that the population at large is at risk of CDI, and there is a real need for intervention and hopefully prevent and lower those numbers of CDI incidents if we were able to develop a vaccine and implement a vaccine. Because I think if I want to leave you with anything today is that vaccines are not the primary driver for reducing disease, but it's actual vaccination. Vaccination, the act of receiving and acknowledging the receipt of a vaccine and accepting vaccination to prevent disease is really what's hopefully going to reduce that yellow, red, and green bar and the all ages bar of blue, to reduce that numbers to hopefully down to 0 if we could. So let's go to the next slide and see how we might be able to do this. Now, it is important to understand that in clinical development, we're not talking about assurances in here. We're talking about a process that is very iterative. It's a process of discovery and research. And I told you, we try to understand how the toxins that this bug produces really interact to create disease so that we can understand the mechanisms of how we might block those and how we might neutralize those responses so that we have an opportunity to stop the disease before it happens, hopefully, before that infection takes place, and the ability to continue to destroy the lumen, the CDI lumen, and cause infection and all the causes that you see in serious CDI. So what we do is we look to understand the bacterium, and in particular the toxins for CDI. And these toxins are really the effector or the mechanism by which this bacterium has an opportunity to grow and develop. So if we understand these toxins well, and you can see there in the picture, we understand that there is a site, the red sites appropriately of cell destruction. This is the nuclear or the string of protein of amino acids and proteins that are able to cause cell destruction and cell entry. We have a binding domain in there as well. If we understand and are able to utilize that to our advantage, we can design a vaccine that will help prevent binding, that will help prevent cell entry, and that will help prevent cell destruction ultimately. So we know that this is possible because historically we have toxin-based vaccines that work in exactly the same way. We have examples like diphtheria, cholera, and tetanus, for which we still receive vaccination today, where these vaccines prevent the toxin from entering cells, from causing the cell destruction, and therefore they're considered vaccines that are antitoxin, meaning they elicit antibodies that will block those toxins, and that's their what we call mode of action. So for a future CDI vaccine, what we hope for and what we are testing for is the neutralization of toxin A and toxin B. And we know so far that we are able not only to produce this toxin, we are able to elicit the response, we see antibodies. Now, what we don't know yet is it effective? Will it work? Do we have the right mechanisms? Did we design the right vaccine? So in our next slide, I'm going to show you how we might test for those. And this is the stage where we are. So Christian, next slide, please. So currently today, we are running one of the largest efficacy trials called CLOVER, which stands for Clostridium difficile Vaccine Efficacy Trial. Now, across the world, we have an incredible participation of 17,536 subjects who are enrolled across 397 sites and 23 countries. Now, think about it. These individuals are healthy individuals of age 50 and older, and they are committing to working with us to understand if they have diarrheal event, to send us their own stools so that we can collect, we can test for CDI. So we're looking for diarrheal stools that fit a certain recommendation with the current recommendations. So we can really diagnose and test if CDI was the cause of those infections. Now, by comparing the groups who received the vaccine versus the group who didn't receive the vaccine, the placebo arm, we are then able to see if the vaccinated group has benefited from a protective effect of this vaccine. And that's really the simplicity of this, while also its complexity. We rely on a large group of individuals to really tell us from their diarrheal experiences to inform us on how, on all the cases of diarrhea they might be having, so that we then have ability to compare. And this is a great effort, and I want to say a big thank you to all 17,536 subjects that are enrolled today. This is by no means an easy study to participate in. It's a long study. We are now on our third year and continuing, but hopefully in the near future, we will be able to determine whether this vaccine may be protective and what level of protection might it have in a population which is at risk of contracting CDI. And that's really important. These are individuals who have had antibiotics, who perhaps have contact with the health system, who are in the community, and they are helping us understand and develop this vaccine in partnership with Pfizer. Next slide, please. Now, one more thing that Pfizer is taking great pride is to work with institutions such as the Peggy Lillis Foundation, like we are here today, and others, to really commit to not only understanding how the vaccine may play an effect, but also to understand the needs for education and awareness. And that's really important. I talked about vaccination versus vaccine, the importance of understanding the commitment to vaccination, understanding why it is important to take that step and get yourself vaccinated against the disease. So we are working with a panel of multidisciplinary experts across the area of C. difficile to really understand what else could we do to help continue the education and awareness at CDI. Because only by you knowing what you might be dealing with can you take action. And that's really at the core of what we are talking about. So this is launched at ID Week last year in October 2019 with an initial C. difficile awareness group, which is a panel of members, and we meet quite regularly to share information and to identify opportunities to continue to raise awareness. And it's really with that one voice, with an urgent need to gather the many voices and really call to action, we hope to increase the awareness of C. difficile by now trying to help individuals get the best out of what is available today, but hopefully in the future continue to help everybody understand what options they have, what can they do to prevent and treat this disease as we look at CDI and hopefully help reduce that burden of disease that we saw earlier in the presentation. So next slide. So key takeaway points for you. CDI, as you understand it, is a potentially life-threatening prevalent medical disease. It exists quite prevalently across all ages, but in principle, in majority of cases, in the older population, for its many reasons, for the fact that we are all aging and we are all more susceptible to CDI. Now, Pfizer's vaccine candidate, and candidate is an important word here because it's not yet proven, so we are still studying it, is continued to be evaluating for the prevention of CDI, and in particular, the prevention of primary CDI. So we are in a phase 3 efficacy study, which is called CLOVER, and we have a fast-track designation since August 2014 by FDA. Because really the FDA, the regulatory body of the US, plus the CDC and this group here, we all agree that there is a large unmet medical need. Now, as Pfizer, we are committed to working with individuals and to continue to prioritize our efforts and recognize the important seriousness of this urgent public health threat, and hopefully help develop future preventative methods that might help reduce CDI infections in the future. So I want to leave you with these thoughts. I hope this was valuable, and I hope that this spiked your interest in the future for a potential preventative tool if we manage to get it right. So I'm really excited about it. I hope you are too. So thank you, Christian. So if the vaccine is successful, the indication will point to individuals who are 50 and older for prevention of primary CDI infections. So together with your doctor, you'll be able to discuss whether these are the right options for you. We are aiming to make it as available as possible by working with the CDC in terms of the recommendations that will also drive the ability to have this vaccine supported across the various payers and the various access points such as pharmacies and doctors. So yes, we will aim to make it available, but it will be a decision of your doctor whether it is the right option for you. So that's a really important question, Pam. Thank you for that question. Is it available to patients who have had previous bouts of C. difficile? So it's important to understand the vaccine is crucially designed for primary CDI. This is new infections of CDI. Although we are looking at potential reinfection or potential recurrent disease, what we're really looking to do is prevent that first case. Saying that, the definition of first case of CDI, it really is each infection may be a primary infection, which means that if you had CDI before, it may be that the vaccine works for you if it's not considered recurrent disease, but it's considered a new infection of CDI. So we have a question from Kevin. He's asking what's the proposed frequency annually. Yes, so it's not annual. Right now, the current schedule as it stands, it's a 3-dose vaccine, which may be given once. We don't know yet how long it would last, but we would expect at least a duration of initially 4.5 years. That's what we are looking at by the time that we might be approved by the FDA. Now, like other vaccines, as we continue to develop the vaccine and we continue to understand its characteristics, it may be longer or it may need revaccination like other vaccines in 10 years intervals, 5 years intervals. We still don't know. But it's very likely that it will be a period of time with a 3-dose vaccine given for maximum protection. Now, we understand that 3 doses during 6 months, it's an intensive schedule. So we are also looking at opportunities to reduce that schedule and make the vaccine more flexible for individuals. So you are able to complete that schedule in a much more flexible manner. But clinical studies will tell us if that will be possible. Now, that's a very good question, Kimberly. So when we design vaccines, and as you know, there are what we consider live vaccines or live attenuated vaccines. There are subunit vaccines, and now there will be a whole host of RNA, DNA vaccines, etc. But traditionally, we are either looking at live versus subunit or protein vaccines. In this case, the toxin is not live. Toxin is a byproduct of the bacterium. That's the live component, but we don't have that in our vaccine. We don't have any component of the bacterium. We have toxins, and within the toxin protein, we have neutralized what would cause toxicity. So without changing the structure, the way the toxin looks, we are able to detoxify it, so remove those areas that would cause potentially the breakdown of the cell and the disease, and we are able to generate antibodies that are able to recognize those important aspects of the toxin. So we keep our toxin as natural as possible without it having an ability to cause disease, and that's really key. So it's both genetically modified and chemically modified to make sure that there is no chance of it reverting to a prior state that could cause potential disease. So it's so far presents a very tolerable, safe profile for this vaccine still in development. Now, Nick, that's a question we get quite often. Will it impact CDI colonization? I've mentioned in my previous answer to the question, we're really going after neutralizing toxins, not the bacterium. So we believe that the colonization or the C. difficile in itself would remain behind. What we would be blocking is the potential toxins from affecting disease. Now, if we consider that, that's not necessarily a bad thing. Our microbiome is constituted of many bacteriums, many different entities that are symbiotic. And when we can, we are better to leave those relationships intact so that the vaccines naturally have an opportunity to keep in check with each other. So we believe that actually by just removing that toxin ability, that toxic portion of the causing disease action, we may be able to stop the disease without causing a potential gap in the niche of microbiome that we have. So right now, we would still have colonized CDVI individuals, but the infection would be removed.
Thank you so much, Catia. We appreciate it. So have a great one. I'll talk to you soon.